Next speaker is Layla, weighted distance-based approach for deriving consensus tumor evolutionary trees. Take it away.
Wonderful. Thank you so much. Thank you to the organizers for allowing me to be here today to present this work. Leah gave a great introduction to tumor evolution, so I'll go through this quickly. Tumors, as we know, are the result of an evolutionary process where at some point somatic mutations were obtained, and then different descendants of that original cell may acquire different mutations as time goes on. So we can think about the history of a tumor using two dimensions, one dimension representing time, the other dimension representing different cell populations. And different populations might grow and sweep through or disappear over the time of the history of the tumor. Unfortunately, we don't actually observe this process. What we get is DNA sequencing data, and from a single or multiple time points, we want to be able to infer what was the history of a tumor. So you can think of this as being represented by a particular type of rooted tree, again, where nodes represent individual populations and edges represent ancestral relationships between them. There's lots of reasons why we might want to infer a tree like this. The more we know, the better. What's more is there are potential implications thinking about treating different cancer types. So for instance, if you think about a history that has two branches like this and you treat the mutations on one branch, you lead to a patient that is susceptible to relapse. So if we know something about the structure, it might be able to give us better information putting together individualized treatment plans. So there's been a lot of work on how we infer these trees from different types of sequencing data. This started back with single cell and single sample, leading into multi-sample, and then more recently thinking about single cell methods. This is just a subset of the methods out there that exist. What's more is these differ on different dimensions, as Leah pointed out really great, that some of them use SNVs, some of them are starting to use copy number variations or other structural variants. And so there really is a lot of work going on in here because this is not a solved problem yet. What's more is you get more and more data sets where there might be multiple different types of sequencing data, and so you have to run multiple different methods. And so what this means is it might be common that we have a single patient where we might have several different tumor evolutionary histories or clonal trees that we've inferred. Again, this can come from different sequencing types or just even different algorithms run on that same patient. So there's been recent interest in thinking about how can we combine these to create a consensus evolutionary history that maybe gives us the important information from across different trees. So one method, graphic, which came out of my lab, used a graph-based approach to try to create this tree, and in particular looking for one that minimized a total distance from the input trees to the consensus trees. There were a number of different limitations. It relied on a very simple distance measure, something called parent-child, that only looks at close relationships between parent and children mutations. It only considered a single mutation clustering. It had to be sort of a pre-processing step. And what's more is every single input tree was treated exactly the same. Again, if they're coming from different types of sequencing or different algorithms, we might have more information than just treating them as the same. Another method that then came out, this was out of Mohammed Al-Kabir's lab, generalized this and said, well, maybe we want to look at multiple different consensus trees, but had some of these same limitations. It was stuck using parent-child distance. It only considered sort of non-clustered mutations in there. So again, it had a single clustering. And again, it treated all trees equally. A more recent method out of Russell Schwartz's lab, ConTreeDP, took a different approach. It wasn't doing distance-based. It was trying to maximize the number of directed partitions, but still it allowed you to have multiple different clusterings, but still treats all input trees equally. So my group looked at this and said, hey, let's see if we can do something that allows us to address these limitations. So we're introducing a new method called TULIP that allows us to overcome these. So I've got a very short amount of time for this talk, so I'm only going to be able to go into this in high level. But first, I want to just give you the problem that we're solving. So this is the weighted tumor tree consensus problem. So we're given as input a set of trees, and for each tree, we have a weight that represents something about our confidence in that. So higher weight means we want to weight this tree more in our consensus. We also have an input as a distance metric. And from those, we want to find the median tree that's a weighted median tree that minimizes the weighted distance to all input trees. In particular, we're going to focus on using a different distance measure, something that's got a little bit more information than parent-child. We're going to look at something called ancestor-descendant distance. And so really this is allowing us to look at all ancestor and descendant relationships, not just the parent-child ones. The short version is that it calculates the number of unique ancestor-descendant relationships that exist in one tree but not the other. So this is the problem that TULIP solves. TULIP, I'm just going to go into high level. It's got two main steps. If you're interested in the details, I'm happy to talk to anyone after. So in the first step, we're going to use an integer linear program. And in this integer linear program, we're not finding the consensus tree right away. What we're going to identify is a very particular DAG that has all directed ancestral relationships that will exist in our final consensus tree. So once we have this particular DAG, in fact, it has some special properties, and we can show that the transitive reduction of this graph does, in fact, give us a tree. And so that is going to be our consensus tree that we find. Again, I'm glossing over lots of details in here. But in summary, this is our method TULIP, and it solves that problem exactly, the weighted M tumor tree consensus problem. So just to give you a quick glimpse of some of the results, so we applied this to a triple-negative breast cancer data set. And so what we have here are 3 input trees that were created from 3 different single-cell methods. What I'm highlighting here in yellow is the node in each one that contains this particular mutation, MAP3K4. So if we run TULIP on these 3 trees, we get a consensus tree. You notice I'm running it here with equal weights. And we get the consensus tree that is the same as the physics input tree. So if we run contree-DP, we actually get the same tree on this example. If we run graphic, we get a different tree, and this is relating to the mutation clustering issue, that graphic forces this mutation to be in its own node, and so it ends up sort of in a different place from here. But where this gets really interesting is when we consider the fact that we can actually now incorporate weights into this problem. So let me orient you to what's going on up here. So we've got a ternary diagram in here. And so what this means is every corner is going to represent if we put 100% of the weight on one input tree. So this corner here is if we had 100% of the weight on site, the top one 100% on sci-fit, 100% on physics in the different corners. The star in the middle is showing us what we would get if we weighted all of them equally. So the colors represent different trees or sets of trees that we can get when we have sort of different weighting schemes in here. So not surprisingly, each corner is dominated by the method that is highly weighted in there. But we see some interesting things in that the tree in the center, the physics tree, which we return, we get sort of a much larger space from there. And we can see sort of some boundaries between these different weighting schemes as we sort of see the different clusterings that end up showing up in here. And so it starts to allow us to see sort of what happens as things change as we change our weights. So for instance, any cell that has a number on here is telling us we had multiple trees that were optimal. And so the set with 2 that's close to the center includes that same physics tree as well as one other one. And so you can start to see a lot of really interesting things here. I'm just going to quickly show you 2 other data sets that we ran this on to give you a sense of the different types of things that we might see. So here's one example where anything that's sort of clustered at all, sort of weighted similarly, we're getting the same tree, versus one where there's a lot of different variation going on in here. So I think there's a lot of possibilities for the use of a method like this, and really starting to think about what it tells us about the space of these trees as we sort of add different inputs and the different things that we know about them. So this is work done by Kathy and Matthew, who are actually 2 really bright undergraduates in my lab. Really, this is their project. It's going to appear at ISMB this summer. So with that, I just want to thank the rest of my lab and funding sources and all of you for being here.
Thank you very much. Got time for questions? Question there?
Quick question. The boundaries on those triangles, do they correspond to specific events on the trees, like a rotation or like?
Yeah, so these are just all mixtures of the different confidence weighting schemes. So every point in here will add up, be 3 values that add up to 1. And so oh, you're asking, like, is this like a different sort of, yeah, so this is sort of there's different clusterings for these different trees. And so the boundary is, the fact that we're having multiple trees on there is telling you that you can sort of have different orders that you're changing those clusterings and cluster this group first or cluster that group first. Good question.
Yeah, I guess how did you compute these symbols?
Yeah, so really, we basically did a grid search of the simplex here. Yeah.
I thought maybe you could
Yeah.
get it right from your application.
I saw you.
You see me. I had one very general question, like what would be the practical application of this one if I'm analyzing my data, like what exactly?
Of looking at a plot like this?
What would be the outcome of
Yeah,
applying
so I think the question is, the place to start is how do you weight these input trees? That itself is not necessarily an easy question. You might rely on sort of an expert, but they're going to say like, I have higher confidence in this tree than that tree. So we sort of have like given a couple of ways that if you have a ranking, you can compute weightings. But knowing something about like, all right, if I shift a little bit, do I get the same tree? Versus if I shift a little bit, am I changing? It's going to tell you something about your confidence in the consensus tree and whether you should be thinking about, all right, let me look at these set and look at what's common amongst these set. And I'm going to consider those pieces to be the parts of the tree that I'm really confident in. Versus something like this where you can say, okay, I feel pretty good about, any of, like, the weightings don't change things, so I feel pretty good about the consensus tree.
So it can help in a way to identify outlier maybe.
Possibly. Yeah, I mean, that's another thing, like, there's lots of stuff we want to do with this, but thinking about, like, as the trees on the corners, like, change, like, how does that change the space? Does it sort of say, oh, okay, these trees all really agree, and it's this one problem child here?
I have a curiosity. This is not recorded, right? Like when we introduced the idea of like consensus trees was because we wanted to understand. I sequenced like 100 different patients. What am I seeing, right? So the consensus was not about a single patient, was about 100 people and trying to understand something about the disease and maybe repeats across patients. While here, it looks I'm trying to make happy and different algorithms, right? And say, oh, this one is better than that, and so do I really want to do this consensus, or maybe I care mostly about the consensus across patients?
That's a good question. We've mostly only thought about it in terms of a single patient. I think there are things that, if you're looking at a consensus across different patients, there's certainly not going to be exactly identical things in the tree. You're going to want to identify what are the core components of it.
And that's where it becomes difficult.
Yeah, and so that's not exactly how this is set up right now, but I think some of these ideas might transfer into that same space. Could be really interesting to explore.
Oh yeah, I was just on the application. I was thinking, could you use this to take confidence values from a single tree inference algorithm, and then
so if you're doing it from a single tree inference algorithm, it would have to be something that's stochastic so that you would potentially get multiple different trees from it. Again, this isn't something we've thought too deeply about, but I could see some sort of version of some sort of like almost bootstrapping like type of thing with it.
Yeah, it kind of applies when you have like more mutations.
Yeah,
yeah.
Possibly.
Okay, no more questions. Let's thank Layla again.